All right, welcome back, guys. If you want to post in the chat, you can go ahead and we'll get started. See who all is here. But again, thank you guys for tuning in. I appreciate you guys taking the time out of your day to do so. I'm going to start off with answering a few questions that were presented in your discussion board. Some people had some really good questions that I felt like needed to be addressed. There was one question: what if there were more than one start codon in a gene? And turns out you can actually do that. So met or methionine is just an amino acid. So there's a start codon that will work for the start of a gene. Inside that gene, there could be multiple AUGs or codons that code for methionine. So a protein can have more than one methionine amino acid in the end. That's just part of it. Another good question was, what if a gene had more than one stop codon? All right, so that could be bad because it could lead to a nonsense mutation, that's which we're going to talk about today, or it could also lead to just multiple genes within the gene. So as we talked about before, that a gene can contain several exons that could all have their own stop codons. So it could allow for diversity within a protein from a single gene. So those are really good questions. You guys are doing great work on the discussion boards, so just want to keep that up. I got another one today. The quiz will be posted after the lecture today, and the quiz will be attached to it, so that's good, unlike last time. So thank you for your patience on that. Today, the lecture is not going to be very long. It's only going to be about mutations, and there's not too much to go over. So we'll buzz through this, give you some time to work on that. And then next time, we will talk about PCR and polymerase chain reaction and move into that. And we may also do a forensics lab, which I realize that it's not ideal to do it online, but we're going to do the best we can. It's pretty fun. Some of the lab procedures, they're really fun to do, but unfortunately, we're not in the lab. So I'm going to make the best of it I can for you. All right, so here we go. Today we're going to talk a little bit about translation, just to recap that. Again, we're taking the messenger RNA, which is out in the cytoplasm, and converting it into a polypeptide chain, as we see down here. All right, we're doing that through the process of translation, using the help of our rRNA or ribosomes. We're also going to use the tRNAs, which are going to bring in the amino acids to the messenger RNA. The messenger RNA is the assembly line, the ribosomes are the factory, and the tRNAs are the truck drivers bringing in the parts. And that's where we're going to assemble everything together in the ribosome. All right. So there seems to be quite a bit of a lag today in the streaming. So if you have any questions, just make sure you post and I'll get to them when I can. Hopefully you watched the video and hopefully it made sense. They did a really good job kind of discussing everything from transcription to translation. I felt like it really tied everything together. So if you didn't give this a look, give it a look. Last but not least was polysomes. Many ribosomes may simultaneously translate the same messenger RNA forming polysomes. So as we take that messenger RNA and put it out into the cytoplasm, you may need more than one of these proteins because it's very important. Maybe it's like a hemoglobin. You're making multiple red blood cells. In the end, though, you can look at this electron microscope image. You can see there's multiple different polypeptide chains forming all from the same gene. Okay, this is the same gene. We're making the same exact protein. This is different than the Christmas trees. All right, so that's a discussion board question. I'm going to have you go back if you want to answer that. Go look at the Christmas trees when we talked about those versus polysomes. Take-home message. Translation converts protein building information carried by messenger RNA into a polypeptide. During initiation, a messenger RNA, an initiator tRNA, and two ribosome subunits join together. That's our whole factory. During elongation, amino acids are delivered to the complex by tRNAs in order dictated by the successive messenger RNA codons. All right, the codons code for everything. Termination occurs when the ribosomes reach a stop codon in the messenger RNA. The messenger RNA and the polypeptides are then released, and the ribosome disassembles, and now you have your mature polypeptide chain. Not a mature protein, got to fold it yet. All right, so today, the cool stuff, mutations. You guys did a research project over different mutations that affect the human body or different species, which was really cool. I enjoyed reading those. A couple of those will be actually mentioned today as they are mentioned in the textbook. So, if a nucleotide sequence of a gene changes, it may result in an altered gene product with harmful effects. Now, as we found out, a lot of times, yes, they're harmful. But every now and then, we get small mutations that don't harm us. They don't always necessarily give us great strength or anything like bone claws or the ability to rapidly heal ourselves like Wolverine here, but they do happen. Okay, so mutations are small-scale changes in the nucleotide sequence of a cell's DNA that alter the genetic code. And again, there was a question in the previous chapter or previous video that asked, what if the messenger RNA screwed up and put the wrong nucleotide in? That too would be a mutation only to the messenger RNA, not the DNA, which could still result in a different protein being made, depending on what nucleotide it was. This is a photo. I was trying to look for something more relevant for today, for the coronavirus, the COVID-19. This is kind of a more complex article, so it doesn't do a great job showing you the nucleotide sequence changes for the COVID-19, but as you analyze this photo a little more, basically you get the gist out of it, that there are certain places along the way that this virus has mutated. And as you look across the world map here, you can see that it has changed ever so slightly from Africa to Asia to North America and so forth down the line. So get small little changes and we can kind of track those things and kind of see where they came from. And again, a lot of people were doing science dailies on the coronavirus, and it was not man-made. That scientists went back and looked at this information and they analyzed the origins of the original coronavirus and how it has evolved, and they are pretty certain it is not man-made. So it's not the government's trying to kill us, so rest easy there. They are trying to do the best they can. So hang in there, be safe. Mutations. There's a couple different types of mutations, though, that can occur to DNA. There are silent mutations when the same amino acid is produced. So basically, when you change usually the third position of a codon, it's highly variable. As we go back, there are only 20 amino acids and 64 different combinations, which means, yet again, some of the codons go for the exact same amino acid. Changing that third position usually allows for the exact same one. Not always, but usually. So sometimes it has no effect on the gene or the function of that protein whatsoever. So many of you may actually have silent mutations, which render maybe a protein for our mitochondria slightly different from one another, but it still does the job because it still has the correct amino acids. So kind of a unique fingerprint, you might say, that pertains to all of us. Oh, there's a question here. T asked, what messes up to make mutations? It's not the messenger RNA putting in the wrong nucleotides. If it's not. So I think what she's asking is what messes up to make a mutation. So again, it could go back to the DNA, which could cause a mutation. So during replication, the DNA polymerase could put the wrong one in. So if DNA polymerase could put the wrong nucleotide in, match incorrectly, RNA polymerase could also do that during transcription. So as it's trying to match up, the adenines with uracils and so forth down the line, it could coincidentally put in the wrong nucleotide. Now, that's only going to make maybe one protein or a couple proteins that are screwed up. Once it goes back and transcribes and translates it again, it could be perfectly fine. So you may just have this weird flare-up of bad proteins and then never see it again, just because the RNA polymerase messed it up. But again, we talked about how mutations can occur because of radiation and other things like that. So damage to the DNA can cause those mutations. If that doesn't answer your question, T, please post again. That was a really good question, though. All right, so silent mutations, as you can see in the picture below, you've got the wild type, which wild type, if you're not sure what that means like the normal, just they call it normal or natural thing that occurs out there in nature, so they call it the wild type. You can see the DNA codes in the blue there. You've got the wild type messenger RNA, and that's what it codes for. And then you have the amino acids, which there you go. It's supposed to be met, pro, phen, gly, al, and pro. So then you have the mutated one, and you can see as we go down the stream, that G right there has changed, which affects the messenger RNA, which changed it to a C, but then the amino acids still come out the exact same. It's like, well, we had a mutation. Well, it is again a silent mutation. That third position is highly variable, which means it can lead to whatever. Okay, if you change the second position of the codon, then you're increasing the odds of it being a missense mutation, which we're going to talk about. If you change the first one, you're pretty much guaranteed that there's going to be a mutation. So, changing the first or second, pretty good odds. Third position, highly variable. You can still get a missense mutation by changing the third one if it's the right nucleotide. Alright, so base pair substitution may result in a premature stop codon, which is a nonsense mutation. So as you're going along, you exchange maybe this G for a C. Well, now that could result in a different codon, which now is a stop codon. So this gene that was supposed to code for, say, 1,000 amino acids now only codes for 2. That's not really going to help you at all. 2 amino acids folding together are not going to have the same effect as 1,000 of them folding together. So big difference there. If you get just a different amino acid, everything else is exact same, but you change a phenylalanine to alanine, that's a missense mutation. It will fold slightly different, but it may be close to what you wanted, but slightly different. And that's a classic example of sickle-cell anemia. Changing one nucleotide results in a different structure, which causes it to fold differently, which causes some problems for those individuals, which we will discuss. A deletion or insertion, those are pretty dangerous because once you do that, you either add an extra nucleotide, as you see here, to the code, an extra G, which then affects the codons. So the codon was supposed to be CCA, now it's CCG. Now the next codon should have been ACT, now it's AAC. You can see how drastically it's changed it. Again, we said if we changed, the second nucleotide, we're going to get a mutation, a big one. Well, you didn't just change the second nucleotide for the second codon, you changed the first and second and possibly the third all the way downstream, and that's pretty bad. Deletion does the same thing. You pull this T out, now you've affected everything downstream, which has shoved everything over, which has messed up all the codons, changing the first, second, and third position. You've changed not one amino acid, but several hundreds of them possibly. And this is a classic example of thalassemia, which is a genetic mutation that a lot of people did do in their reports. We'll talk more about that in a minute. Okay, so inversions, these are kind of cool. They do happen where chunks of chromosomes actually break apart. Got another question real quick here. It says, person asks, so UCU to UCC doesn't do anything to the organism? Most likely no. If you go back and look at that amino acid chart, let's see if I can find it. The amino acid chart will show you that right here. If we look at UCU right there codes for serine, and then UCC also codes for serine. They're both right there. So in the end, inevitably, there are silent mutations. All 4 of these code for the exact same amino acid. In fact, there's actually 6 serines, so you can actually change a number of things and still get a serine, but not too much. Proline, the same thing. You get a couple right here. CCU, CCC, CCA, and CCG all code for proline. Every now and then, like I said, you get some of them that are very specific, like methionine, this one, lysine. They all sometimes are very specific, but yeah. All right, another question here. T asked, if a whole strand was messed up like this, what kind of change would that make to a person? If the whole thing was messed up, the whole strand was messed up, like beta thalassemia, for example, that could definitely cause some problems, depending on what gene it is. That's a big factor there. But if that's in every single one of their cells, that is a huge ordeal. Why? Because it's going to cause some health problems. Most of these genes that you have in your body are pretty important because they they function correctly. They've been cultured over time to engineer you and to make you work properly. So without that protein, you're missing a vital part of life or functions that you need. So just again, to answer your question, T, it comes back to what gene it is. But most likely it'll render some big problems. And thalassemia, which we're going to talk about, does cause some problems for the individual. Okay, really good questions, guys. Appreciate those. So going back to the inversions, you have the paracentric and pericentric, which the paracentric at the top here, when only one arm of the chromosome is inverted and does not include the centromere. So on a chromosome, you can have whole chunks or pieces actually just break off, then flip around, and then there you go. So as you look here, you've got the normal chromosome, it has a break, and this is a paracentric because we know because it's not part of the centromere. It will snap, and now the code should have read A, B, C, D, E. Now it reads A, D, C, B, E. Big difference. So sometimes that can cause some mutations. Sometimes it does not, depending on where it breaks at. Pericentric is when the inversion occurs near the centromere, for example, right here in the middle. You can have a whole chunk move and flip. And that too can cause some problems as well. So sometimes you can have specific genes here and specific genes here, and then you go to mix them, you can create some problems there too. Hemoglobin, again, we've got this. We talked about this in the past. Hemoglobin is a protein that binds oxygen in your red blood cells, and it carries it once again from your lungs to all your cells in your body. Hemoglobin molecule consists of 4 polypeptide globulins. They're folded with an iron, that once again, that heme group, which we talked about as a cofactor. So this is the normal hemoglobin, what it looks like. But then when they have the sickle cell, they clump together and they create this sickle shape. So we get this donut shape, or we get this sickle shape. The defects in the polypeptide chains can cause anemia, okay, and affect the red blood cells. So this is just a number of photos that we're going to talk about here. Again, this is the hemoglobin. The blue represents the alpha globulins, the green, the betas, and the red is the heme groups that actually are the cofactors of the iron that helps bind that oxygen. Here, it's just showing you the normal messenger RNA at the top. It's numbered 16 through 30. Then you can see the amino acid sequence here. This is the change for the hemoglobin from normal to sickle cell shape. One nucleotide changes one amino acid. So again, we've got a codon and then another codon, and we change the second position, which I said, again, if you change that second position, you'll probably get a new one, a new amino acid, and we did in this case. So just a simple change, one nucleotide. This is a deletion where a nucleotide has been pulled out, which has again affected every single amino acid or codon downstream. This is like the beta thalassemia. Again, this can cause some big problems. This is another version of beta thalassemia where you have a insertion where you add an extra and it affects everything downstream. Now, if we go back, this deletion or insertion here was caused upstream, which caused many of them to be affected. Here, this one may be towards the end, and as such, it only affected one, but it could still affect a number of them downstream. So sickle-cell anemia is caused by base pair substitution, which again affects red blood cells. Here's the scientific name, the sickle hemoglobin HBS. Once again, cause them to stick together. Over time, sickle can cause damage to organs and even possibly death. Not necessarily is going to cause immediate death. It'll cause a death in the individual over time. But it can clump into capillaries and stuff like that. This is T-Boys, I believe it is, from the music group TLC, probably a little before your guys' time. She's a spokesman for sickle cell anemia. She has it herself, but she has both traits, the sickle and the normal one. So, another question here. What takes an amino acid out in or deletion? So what, like, the amino acids usually aren't removed per se, but is the nucleotides sometimes that are removed or added. What would actually cause a nucleotide to be added? It could just be an extra one by chance. There's also these transposable elements we're going to talk about here in a second. Sometimes the deletion is it just gets cut out, like it just gets forgotten to be put in there, and as such it just skips over it and starts putting stuff in there. Sometimes radiation, which can cause the breaking in between the phosphates and the sugars, and they can actually break off, and it tries to repair it by crunching it together for whatever reason, which is bad, and it kicks out the other one that's causing a kink. So a number of factors can actually do that, cause an insertion or deletion. The Thalassemia and Frameshifts, another type, once again, affects the beta globin gene, causes the reading frame to be a frame shift. Frame shifts garble the genetic message, just as incorrectly grouping a series of letters garbles the meaning of the sentence. So if you took out one letter of a sentence and you crammed everything over in the slots, it wouldn't make any sense. Kind of like an encryption. For those of you who did Code Busters for Science Olympiad, you know how frustrating that can be. So thalassemia and transposable elements. Here's to answer your question a little more, T. Beta thalassemia can also be caused by insertion mutations, which also cause frame shift. Insertion mutations are often caused by the activity of transposable elements, which are segments of DNA that can insert themselves into a chromosome. And you're like, where do these come from? These nucleotides naturally exist in your cells. So you're trying to replicate DNA and whatnot. Sometimes they can be from damaged or broken chromosomes. Sometimes they just stick together for whatever reason, and they're just floating around. And then they just get jammed in there because of radiation, weak bonds, whatever it is. Now, what are the odds of this? I mean, this is kind of low, but does it happen? Yes, it does happen. Usually your polymerase can find these things, cut them out. But again, if it doesn't know what was original and what wasn't, it can cut out the wrong things, which can cause a permanent change. So if this happens to one cell, it may not be that big of a deal because now that cell may die off, or it could be a big deal or cause cancer, or if it doesn't cause cancer, you're talking about a localized group of a couple cells that just don't work right. So it's not that it's going to hurt your whole body. The worst part would be when it affects the entire organism, like a gamete. That would cause a problem, egg or sperm cell, for the individual. Okay, I believe that is pretty much it. Okay, mutations alter proteins based on the DNA or even the messenger RNA if that was the case. Premature stop codons cause nonsense. Sometimes if you just substitute one out, it can cause missense. If you have frame shift, those are pretty bad. They can mess up everything downstream, insertion or deletion. All right. If you have any questions, go ahead and post in there, and I'll answer them as quickly as possible. But again, thank you guys for tuning in. I really appreciate that. Hopefully you're all staying safe and healthy, and so are your families. After this is over, I will post the discussion board. You guys did a really good job. Again, I encourage you to try to come up with more opinion-based or what-if type questions to stimulate further conversations, because otherwise you do come up with the exact same answers. I tried to make mine more opinion-based, so that way you can get a plethora of answers going forward. There's also going to be the quiz. I double-checked it. It is attached this time, so you should be able to take that real quick. And that's just to guide you to see what you learned and what you didn't learn from the video or the lecture. So if you missed something, just go back and take a look at it. All right, I don't see any questions. I'll give a few more minutes here, but we'll go from there. If you guys have any feedback regarding the videos or things that you would like to do over this extended break, just email me and I'll be glad to review those items and go from there. All right, not seeing any questions, so I'm going to turn you loose. Again, check out the Google Classroom. Thanks for tuning in, guys. Have a great day.